Welcome to the MOOC Interactomics course. In our last lecture, we studied that the major strength of surface plasmon resonance SPR biosensor is their versatility and ease of use. The SPR platform allows the ligand analyte interaction for a wide range of molecular weights and varying affinities and compatibility for small molecules dissolved in organic solvents. These biosensors play a very important role in biological research to monitor macromolecular interactions in real time without the need of labels. It is one of the reliable platforms to determine affinity, kinetics, concentration analysis and immunogenicity for a variety of interactions, which we are going to discuss in today's lecture. We will also learn a little on how to analyze the data and perform data analysis using software. Let us now continue our discussion with Mr. Lalit Kishore to discuss various applications of SPR. So, Lalit, welcome to the discussion on SPR technology.
Thank you. Going on to the next application, I always think that concentration is very less studied, but it's very important, is because if you look at concentration analysis across the world for proteins, there is no way that someone can measure active protein concentration without having calibrants in their hands. Right. Whenever you give a student a protein and say, please measure the concentration, the first thing that he will ask you is for calibrants. Right. He will ask you for standards. And the problem with these standards is that sometimes they are not available. Sometimes they are extremely expensive, and being proteins, like you know, sometimes they degrade.
Not very stable. Yeah.
They're not stable. So there is a great need for having a concentration analysis technique that does not need calibrants. And that's where Biocore comes in again. Biocore does something that's called CFCA, which is calibrant-free concentration analysis. And so within 5 minutes, if you have a specific binder for a protein, you can actually calculate the concentration of that protein without the need for calibrants. And since you already seen in screen 1 where we talked about specificity, we are talking about specific binding. Right. So what is measured is not just total protein concentration. What is measured is specific active protein concentration.
I think that's a very strong application of it, because many times you'd like to know how accurately you can determine the protein concentration.
Absolutely, especially in quality control and in filling in biopharmaceuticals again, where people need to exactly estimate how much they are actually filling in the final vial that actually goes to the patient. They need more accurate methods of measuring active protein concentration. And that is where I think Biocore will play a very big role in letting people estimate that active protein concentration. Now, once you've seen concentration, the next thing is immunogenicity. Worldwide, with the increased biopharmaceuticals, drugs everywhere need to be tested for immunogenicity. Right. Immunogenicity is about direct measurement of anti-drug antibodies in the which should be measured in serum. Mm-hmm. It's also about bringing a regulatory framework into a system. Right now, we talked about only technology and science, but suddenly when it comes to drugs, regulators come in. Right. So can we actually accurately, confidently measure anti-drug antibodies in animal sera or in human sera at clinical trial levels? And Biocore can actually be used for accurate measurement of anti-drug antibodies and for immunogenicity testing of biopharmaceuticals. So that's another major application of Biocore as it comes in. The last application of Biocore, which if you remember when I talked about in the first slide about specificity, now if you have a heterogeneous mixture that is flowing over a ligand and something within that mixture is actually bound, and you see a curve and you know that something has bound, but you don't know what it is that is bound. So you can use a technique called SPRMS, where you can take the bound analyte, separate it into a vial, and then actually take it to a mass spec and identify the protein. So in addition to doing all that it did before, now you can actually also find out what is it that is bound.
So definitely it's very important, because many times you will not know like what the unknown target is, which is interacting or binding, right?
Yes. So if you don't know, if you have this unknown target, for example, if you have a receptor and if you have cell lysate, right, or if you have some kind of a homogeneous tissue lysate, which you are flowing over the ligand, and now you can actually find out what that is. So it is used in applications like ligand fishing, for example, where you're fishing for a ligand. So those are the kind of examples that.
But in MS, you will definitely require protein above certain threshold, right? So how you overcome that issue, because the binding will not
be minimal.
Enough protein.
So one thing is that this is a very small interaction happening, so the amount of protein that you collect may not be sufficient. The only way you can overcome it is by actually doing it multiple times, collecting enough so that you can actually get some kind of an MS response. And that is what most of our users do, is that they run the same binding assay about 10-20 times and collect the bound analyte and then take it to an MS and then get their result.
So in Biocore you have a way to collect the flow in the flow cell.
Yes.
And then actually you can accumulate that in multiple
runs and then take it.
Then concentrate that and then you do further characterization.
Yes. So just to summarize, again I will just go back to my the first slide that I showed here, which is here. These are the 6 things that a Biocore can be used for: specificity, kinetics, affinity, concentration, immunogenicity, and mass spec analysis. So those are the 6 broad applications of Biocore SPR technology.
I think those are very important, because many times a good decision about these products beforehand, about characterizing these proteins, can save you a lot of money, a lot of effort down the road, because
absolutely,
if you can do those experiments in the beginning.
Most often when we actually talk to pharma industry, we tell them that Biocore or SPR technology is not for success but more for failure. Right. Only thing is that we say that it is for early failure and it is for cheap failure. Right. So do not spend too much money on something that does not work. Right. Might as well fail early. Right. So that is the basic reason why SPR should be used.
I think that's very important. So can you briefly tell us about what are the major instrumentation available from the Biocore technology currently for doing the surface plasmon resonance based experiments?
There are basically 4 different Biocore instruments available. There's a very small Biocore which is called the X100. You can see it on my pitch. It has 2 flow cells and it can do some beginning analysis. And then you have the Biocore 3000, which is an academic favorite. A lot of academicians like it. A lot of customers in India who are academics have the Biocore 3000. Then we have the latest, which is the Biocore T200. Very special again because it has all the things that the Biocore 3000 has, but it is regulatory approved. So if you are a company that works with FDA, that works with EMEA, that works with DGCA or some of these regulatory authorities, then I think that you should be using the Biocore T200. The Biocore 4000 has 20 different immobilization sites on it, and Biocore 4000 can be used with multiple 384 well plates, with robotics. So if you're a company that has extremely high throughput screening, if you're a company that does a lot of immunogenicity experiments, or if you're doing batch testing or release testing using Biocore, then you should be using the Biocore 4000. I must say that most of the customers in India use either the Biocore 3000 or use the Biocore T200.
So how easy it is to do these SPR experiment, and especially the kinetic analysis by using some software available from Biocore?
The most important thing in a Biocore experiment is actually the experiment design. It's very easy to do the analysis, and let me just show you an example of a typical result. Okay. And let me show you how the analysis works out.
Sure.
So if you can see my computer, so I'm going to open a typical Biocore result, and here is a Biocore result. This is a Biocore result where 5 samples of different concentrations were run over a fixed ligand which was on the chip, and now I'm trying to do kinetic analysis. So if you look at these 5 results, I select them and I show the results, and these are the results. Now this is what a typical result looks like, and there's nothing to be worried about because it looks odd. The idea is that your results are all embedded somewhere here. These 2 big peaks that you see are regenerations. So the first thing that I do when I do the analysis is actually select the regenerations, which I don't need, and cut them out. So I cut them out here. So I say cut, and then the rest of the results.
So you want to adjust your response axis.
So now I have 5 different concentrations, 1, 2, 3, 4, 5 in different colors, and you have this is the association phase, and this is the dissociation phase. Now I'm going to just baseline these results. So I select the baseline here, and then I just go to the adjustment of y-axis, and I say 0 at the average of selection, and then I say add as new. And now all my 5 results are shown here. Now I'm going to do a quick kinetic analysis, and it's really extremely simple to do a kinetic analysis, because all that I do is say calculate, I say kinetics, simultaneous K on and K off, and I have already done the cutting and the Y transformation, so I say next. And then if I want to, I can go and adjust the start and the end time. So I can actually move this to adjust the start time and the end time of association and dissociation, which I sometimes do, but I think this is pretty well picked up by the software already. So I don't need to do much. I say next, and I enter the different concentrations of each of the samples which were run. They're entered here. Now this is very important.
These concentrations are known concentrations
which you ran
already
on,
started.
which you actually made the concentrations of the analyte and then ran them over the ligand.
Sure.
So you would do that. Now one of the things that is important here is you choose the model. So preferably, it is always better whenever you do characterization, the more you know about your system, the better characterization results you get.
Right.
So in this case, suppose you do know that it's a 1-to-1 binding, then you would choose that binding. But if you want to, you can actually change the binding model. It could be a bivalent analyte, it could be a bivalent ligand,
Right.
it could be a heterogeneous ligand. So depending on the model you choose, so you choose the model that you want to here, and then you say fit. And what happens is curves get fit and the results are thrown up. So your K on and K off are displayed here. So it's as simple as that. So all you need to do is take a ligand, immobilize it on a chip,
Right.
run about 5 different concentrations of your analyte over the chip, and this simple and each of these results, if you look at the x-axis carefully, it's 0 to 600. That means each one of these runs, the entire run
Right.
was 10 minutes,
Yeah.
and you ran 5 samples, so it was 5 into 10 minutes, so 50 minutes, with the time taken in between the runs about another 10 to 20 minutes, about 1 hour 10 minutes, 1 hour 20 minutes, you have the results, and you have already characterized your results because you have the K on and K off calculated. And you can also quickly check how good your results are by quickly checking the residuals, and you can see the residuals here. The chi-square values are really between -5 to +5%. So extremely good
Very tight.
fitting reactions and a very fast analysis that gives you K on and K off.
Yeah, I think it's very extremely fast and very good. way
Very
of
easy to
analyzing
use, very easy to use
interactions.
software for your analysis.
So yeah, it was very useful and informative to see the analysis, like how easy it is to perform the kinetic analysis. Can you give some specific example of doing kinetic analysis by using Biochemical system?
Let me show you one example, and this again goes back to why kinetics is important. Kinetics is extremely important in Biochemical analysis. And let me show you an example. So here's a pitch, about 3 slides on kinetic and affinity analysis using Biochemical. So if you look at this slide, now here on this slide, you have 3 interactions which are captured. You have interaction 1 which is captured in blue, interaction 2 which is captured in red, and interaction 3 which is captured in black. The important thing about all these 3 interactions is that all these 3 interactions have the same affinity. They have varying K on and K off, but they have the same affinity. And this is important in drug discovery because let us say, for example, you were looking for pain relief. If you want pain relief, you want a drug that acts fast and that stays on for a very long time. But if you want a sleeping pill, for example, you want a drug that actually acts slowly but stays on for only a reasonable amount of time and comes off fast enough. So kinetics is very important in choosing a drug candidate. Now this is an example where if someone chose just on the basis of affinity, all these 3 would have been the same. But since they would make their choice based on kinetics, they can actually decide based on the K on and the K off. Now here is a real life example in the next slide which I show you, which is a publication from Amgen and from David Mishka of University of Utah, where actually Amgen uses this data directly to do their clone selection. So they're actually doing MABs selection of maps, selection of clones for maps. And if you look at this, so clone number 1 and clone number 2 have similar affinities, but if you look at their on rate and off rate, there's a tenfold difference. So if you look at these two clones, for example, which clone should they go for? They should go for the clone which is having more kinetically relevant properties. If they use only affinity for making that choice, then there is no choice at all. They're both the same.
Right.
So this is a live example of where kinetics data is actually being used to capture information regarding K on and K off, and then make a more educated, knowledgeable decision on which map to go forward with.
Right. I think it's very interesting to see this, because you will feel that if you just rely totally on the KD values, right? If you do not go individual on the K on and K off.
Yes. So distribute that KD value into K on and K off and make a more knowledgeable decision. Yes.
So it was a very interesting example to appreciate the power of kinetic analysis, right? Can you just brief us about what are the major limitations or shortcomings of various surface plasmon resonance based technologies?
Absolutely. So like any technology, this technology should not be viewed as a silver bullet. It does have shortcomings. One of the significant shortcomings being that if there is any structural differentiation in the protein, this would not be able to capture it. It's a mass-based sensor, so any structural changes will not be captured. There is also a problem that there are situations where you are unable to immobilize the protein that you have onto the chip.
Right.
So you might have to use some capture techniques to do that. There is also this big question about, so you answer these questions about interaction, you answer questions about whether the interaction happens or not, how fast, how slow, how strong, how much.
Right.
You say whether it is safe or not with immunogenicity, and you know what is binding. But then sometimes you want to ask the question, why is it binding?
Right.
And why is it binding is answered by structural studies or thermodynamic studies. So that is where I think Biocore can give you a little bit of a direction, but I think you should do more of an NMR study, or you should do microcalorimetry, and that's what will probably give you more answers on why the interaction is happening.
So further in-depth studies will be required on those
Absolutely.
interactions.
So when you have an interaction happening, and when it's happening very fast, why is it happening fast? Is transition state thermodynamics. So you will have to ask questions in thermodynamics to get those answers. So sometimes further study is essential, and Biocore kind of stops with these applications.
Would you have any final advice, take home to the
Absolutely.
students?
So here's basically 3 rules that we have in Biocore. The first rule in Biocore experiments is Biocore technology is extremely easy to understand. Biocore technology is extremely easy to analyze. A lot of time must be spent on experiment design. The first thing that I would do if I were working with Biocore is spend a lot of time on very carefully considered experiment design. The second thing that I would do if I were conducting a Biocore experiment is that I would make sure that I have an extremely pure ligand that I will put on the chip, and that is extremely important. The last thing that I would consider, and this is true of all experiments, and I'm sure you would agree with me, is that it's garbage in, garbage out. So make sure your sample preps are correct, and remember that this is an analytical instrument. Unlike many other techniques in biotech, this is an analytical instrument. It is a mass sensor. At the end of the day, you could call it an extremely sophisticated weighing machine. So if you put something on it, it will give you the weight. It's as simple as that. Only thing is you have to do it right. So I would say that make sure that your sample prep is perfect, your experiment design is perfect, make sure your ligand is pure.
So I think you very rightly mentioned that a good experimental design, identifying the good ligands, working out the chemistry for immobilization,
Absolutely.
as well as doing the very good sample preparation. All of these are very essential component for doing any successful proteome experiment, and especially the surface plasmon type of experiments.
Yes.
So thank you very much, Lalit, for discussing about the Biocore technology with us today.
Thank you.
And I hope it is informative and useful for my students.
Thank you. Thank you.
Through our discussion on surface plasmon resonance technology, we learned that there are 3 major advantages of this approach. First, the kinetic measurement in real time is the biggest strength of this method. Second, monitoring the adsorption of unlabeled analyte molecule to the immobilized ligand surface, and third, its ability to detect weakly bound interactions due to high surface sensitivity of SPR sensors. In summary, in last two lectures, we learnt about SPR technique, its applications in different fields of research, and a little overview on data analysis. Thank you.